About 2.5 billion years ago, the Earth's atmosphere was very different than Earth's atmosphere today. It was an anaerobic atmosphere, which means that it lacked oxygen, until an ancient ancestor of cyanobacteria started photosynthesizing. The ancestor of cyanobacteria was thought to be what helped create an aerobic atmosphere. The ancestor of cyanobacteria was engulfed by a eukaryotic cell and eventually became the chloroplast. But what practical uses does cyanobacteria have today?
Cyanocystis, a type of modern cyanobacteria, has recently been modified to create complex organic molecules. Hypothetically, these complex organic molecules can include those used for cancer treatments, which are normally made by extracting complex organic chemicals from various plants. Cyanobacteria are cheap, require few nutrients, and can be optimized to produce secondary metabolites, which are byproducts of metabolic pathways. These secondary metabolites can often be used in a wide range of drugs, but are often hard to synthesize on an industrial scale. This study used cyanobacteria as their medium to test cyanobacteria's potential in future industrial drug production. The researchers chose to use the protein cytochrome P450, which is widely noted for its ability to perform difficult oxidation reactions, and the Durin pathway, which has several useful secondary metabolites. To implement the Durin pathway encoded in a plant's CYP79A1 gene, the researchers extracted the gene from a plant and inserted the gene into Cyanocystis plasmid with a lac operon. They then inserted a chemical that acts as a transcription factor, causing the cell to produce P450, effectively activating the process, which they called induction. To implement the Durin pathway encoded in a plant's CYP79A1 gene, the researchers extracted the gene from a plant and inserted the gene into Cyanocystis plasmid with a lac operon. They then inserted a chemical that acts as a transcription factor, causing the cell to produce P450, effectively activating the process, which they called induction.
To test their hypothesis, they inserted the Durin pathway into 2 samples and induced one at 24 hours and recorded the amount of Durin produced at 12-hour intervals. This is shown in Figure 1. Figure 1 shows that the Cyanocystis that was induced created 5.1 milligrams per liter of Durin after 72 hours, and the non-induced Cyanocystis created 1 milligram per liter. Since the induced operon created more Durin, the researchers concluded that Cyanocystis was using the engineered pathway to produce the molecules. Consequently, we can conclude that Cyanocystis could be a viable option for medical macromolecule production on an industrial scale.
If manufactured on an industrial scale, cyanobacteria could have various applications. For instance, the cancer drug Taxol is currently extracted from a chemical in the Pacific Yews bark. By copying its metabolic pathways, cyanobacteria could produce these drugs, so no more deforestation. Similarly, bioplastics could be produced by fixing carbon from the atmosphere without using the traditional petrochemicals, whose extraction and processing causes global warming and deforestation, among other problems. Fragrances and flavoring agents could also be created using the same process, by using cyanobacteria to synthesize various compounds. But why is this process better? Generally, these different chemicals are harvested from plants or created using petrochemicals, which can be very expensive inputs since they are non-renewable, whereas cyanobacteria just need to be modified and bred so that large quantities can be used to synthesize the chemical components. By cutting out the energy-intensive process of synthesizing these molecules using traditional means, we can cut out the expensive byproducts such as carbon dioxide and inputs such as large amounts of energy. In conclusion, through these metabolic engineering techniques, we can save energy, trees, and the planet.